Thank you, respected speakers. My paper is on SMS strategy for molecular diagnosis of retinoblastoma. As we all know, the diagnosis of retinoblastoma is clinical. Radiology provides presumptive diagnosis, and histopathology can give a definitive diagnosis post-enucleation. So sequential molecular screening of RB is available only in few centers and is very expensive, which remains a bottleneck. Hence, we aim to develop an indigenous and cost-effective method for the molecular diagnosis of retinoblastoma. The methodology we used was DNA is isolated from the blood and tumor samples wherever available using the suitable kits. The quantity and the quality of the isolated DNA is checked by the qubit fluorometer and the agarose gel electrophoresis. Sanger sequencing and multiplex ligation dependent probe amplification, that is the MLPA, is used to detect the point mutations and the deletions or duplications of the RB1 gene. Both the steps involve the amplification of the gene or the probe that binds to the gene, which gives us about 90% sensitivity. In addition, we do methylation-specific MLPA for detection of the promoter methylation in RB1 gene, which gives about 95% sensitivity. Next generation sequencing methods for the targeted whole exome sequencing were also done to detect the single nucleotide variations, that is the SNV, and the copy number variants in additional genes other than the RB1. Library preparation using the specific kits was done in-house. Data analysis was done as it is very critical to identify the suitable variants, which are population specific, person specific, structural specific, and functional specific. So this is the channelized flow of the sequential molecular screening of retinoblastoma with integration of data and functional analysis. And this is the flow. And with all the methods and high throughput instruments in place, we complete the analysis starting from sample collection to report generation in just 3 weeks. So genetic analysis and counseling has benefited 543 families with retinoblastoma. That is, we've analyzed 2,120 samples. Patients with mutations, that is germline, were 305, forming about 56%. Patients without mutations, which was sporadic, forming about 44%, that is 238. And families with mutations was 41, forming about 7.5%. If germline, their future siblings and offspring have to be checked clinically and genetically for RB. If there's no mutation in blood, RB will not be inherited. So more than 100 mutations like nonsense, missense, frame shift, splice have been identified, out of which 20 are novel, and these mutations are being characterized for their pathogenic role. Promoter methylation was identified in 4 patients' tumor but not in blood. This epigenetic event is somatic. They're less likely to inherit the mutation to the next generation. You can see a unilateral RB without any family history was found to have a promoter methylated in both the alleles in the promoter region. MICin is a well-characterized gene of RB. Increased copy number of MICin was demonstrated in several cancers. MICin amplification with more than 10 copies was detected in 3 of our patients, correlating with the severity of the disease. CNV analysis of unilateral RB tumor samples was validated and confirmed by the qPCR. Our next generation sequencing analysis has identified several candidate genes involved in the tumorogenesis. We found the gene modification amplifications in MDM4, K1F14, DEK, E2F3, and TP73. These genes are involved in cell cycle regulation, cell proliferation, and apoptosis. Currently, we are studying the role of the individual genes through cell culture models and trying to identify the suitable drugs to reduce the expression. The impact of this study has been seen for 543 families, where the timely and cost-effective diagnosis have helped them in making informed decisions by predicting the possibility of RB in the sibling, offspring, laterality, and secondary cancers as well. Cost has been reduced from $1,800 to $150 for an entire family, and the time from 10 weeks to 3 weeks, which can further be expedited. Thank you very much.
Thank you, ma'am. You have a question? Sure.
Very nice presentation, Dr. Usha. You have done both the genomic and the exomic sequencing. Yeah. Did you help in any prognosticating the disease?
Yeah, that's what I was...
Because it's quite expensive doing both, right?
Yeah, so it's not within the purview of this, but we have seen that MICin amplifications have had a prognostic indication as well. So we are able to predict and preempt for the sibling the offspring, and also prognosticate if the patients who are undergoing radiotherapy, which is still a part of the treatment regimen, will ever get a secondary cancer, which is quite common with the. Thank you.